So we're going to talk about the GQ pathway, which is one of the three important GPCR pathways. And we're going to start with the ligand binding to the extracellular portion of our receptor, which, as you can see, is a 7-transmembrane spanning receptor that goes in and out of the membrane. And as you can see over here, we have our inactive heterotrimeric complex with G alpha Q now, because we're talking about the GQ pathway, bound to the GDP in the inactivated state. Once the ligand binds and the receptor changes its conformation, the affinity for the G protein changes. We have GTP affinity go up and the GDP affinity go down. So now we see the activated G protein now bound to GTP to the alpha subunit. It's now in the activated state, and the beta gamma subunit are off to the side, which may or may not be doing something important. Now we can see our target enzyme. Remember, the activated G protein has a target enzyme, and in this case, the target enzyme is going to be phospholipase C, which is indicated here, and it's going to be acting upon a phospholipid called phosphatidylinositol bisphosphate, PIP2 or PIP2. You can see where it gets its name. You can see that this is a glycerol phospholipid. There's a glycerol backbone, a diacylglycerol. There's a phosphate linked to an inositol with 2 phosphates in the 4 and 5 position. And the phospholipase C is going to act on this phospholipid. And what you're going to see is the formation of 2 important products. You're going to see a diacylglycerol, okay, and you're going to see a IP3, now a 1,4,5 inositol trisphosphate, which we'll abbreviate IP3. And we need to now look at what are the downstream consequences of this cleavage of PIP2. Well, first, let's focus on the IP3. The IP3 is going to act on a ligand-gated ion channel that's on the membrane of the endoplasmic reticulum, which we're going to abbreviate ER, which has a very large store of intracellular calcium. Now, it's sequestered in this organelle, and once the IP3 binds to the IP3 receptor, which is right here, that channel, it's a receptor, but it's a ligand-gated ion channel, that is going to have a conformational change, and now calcium is going to go down its concentration gradient, and you're going to get an increase in intracellular calcium. Now, that increase in intracellular calcium is going to do a variety of different things that we talk about in other videos. There's lots of important effects, including glandular secretion, muscle contraction, things like that. And the calcium also targets a protein kinase called protein kinase C, which is a calcium-dependent protein kinase. And in some cases, the diacylglycerol also can activate this kinase. And as you've learned in other videos, the protein kinase C has the capability to phosphorylate proteins. It's a serine-threonine kinase, and that protein can either be inactive or active. And that protein then gets phosphorylated, now becoming either active or inactive. So just like we saw with protein kinase A, protein kinase C also phosphorylates proteins, and depending on the protein, the activity can go up or down. What's not going to be shown here is there's some forms of PKC that also can go into the nucleus like PKA and have genomic effects as well. So in summary here, the important thing when you activate the GQ pathway is that you get an increase in intracellular calcium. And that calcium can have its own effects in addition to turning on kinases downstream, which can then affect existing proteins or even in some cases affect gene transcription. Now let's talk about how we turn the pathway off. And just like we saw for the other pathways, it's very similar themes. So one obvious idea here is that the ligand is going to unbind and just diffuse away. Okay, so we can call that number one. Of course, like all of these G proteins, we have the intrinsic GTPase activity, which is going to turn that G protein off by getting rid of the GTP and then reforming the heterotrimeric inactive complex. Now, just like we discussed for the GS pathway where we can metabolize cyclic AMP, there's also enzymes that can metabolize IP3 to IP2 and beyond, and those would be inositol phosphatases, which would be number 3. We also have capability to get rid of the calcium. There's mechanisms that will put calcium back out of the cell or into the endoplasmic reticulum. So we have mechanisms that will put the calcium back against its concentration gradient. We have calcium ATPases that hydrolyze ATP. Remember, we're taking calcium still against its gradient, so it requires energy. This is a primary active transport mechanism. We also have ATPases these are calcium ATPases that are on the cell membrane. We also have mechanisms that have calcium go out in a secondary active transport that utilizes the sodium gradient. We have 3 sodium can come in down its concentration gradient, and that would be a sodium calcium exchanger, which would be, we can just call that NCX. That would still be kind of number 4. It's a mechanism to get rid of calcium. And then finally, the phosphate that was put on by protein kinase C can be removed here. Number 5 is a protein phosphatases that remove the phosphate that PKC put on. And just like we saw for the GS and GI pathways, there's an additional mechanism that we'll discuss separately where you can have downregulation, desensitization of the receptor. It's more of a chronic adaptation that can happen when there's long-term stimulation of the receptor.